Hello everyone, my name is Johnathan Galbreath and this is my project about fluorescence. So what is fluorescence? Fluorescence is the ability of certain chemicals to absorb radiation and emit visible light. As you can see in the picture on the left, this diagram describes the absorption and emission of light. The fluorescence process revolves around three fundamental stages: excitation, excited state lifetime, and the emission. Initially, excitation occurs when particles called photons from visible light are absorbed by a light-absorbing molecule, propelling it into a higher energy state. However, this heightened energy state is transient, and the molecule promptly seeks to release this excess energy, marking the onset of the decay process known as the excited state lifetime. This phase is fleeting, lasting only nanoseconds. Finally, the high-energy photon undergoes a rapid decay, returning to its original state and emitting the excess energy as fluorescence. This intricate interplay of excitation, excited state lifetime, and emission defines the fluorescence phenomenon, crucial in various scientific and technological applications. Luminescence is also generated when molecules are excited by ultraviolet or visible light, resulting in the emission of light from their electronically excited state. As you can see from the graph, there is a peak emission at a wavelength of about 575. In this video, I will go over how different wavelengths of light impact the luminosity of fluorescein. As you can see from the data collected, there is a peak luminosity when the LED bars were shining light blue. Hello everyone. Before I get started with this experiment, I want to do a brief overview of all the materials present in this experiment. The first material and crux of this experiment is fluorescein, which I have right here. This will be poured into a 10 milliliter graduated cylinder. The amount of fluorescein used in this experiment will be 4 milliliters. It will then be diluted in a beaker of 250 milliliters with roughly 150 milliliters of water. The two LED bars that are present will provide a variety of colored lights which will affect the illumination of fluorescein. I will then use a luminescence meter, which I have right here, to measure illumination of fluorescein corresponding to the colored LED light. Before I perform the experiment, it is significant to see that non-diluted fluorescein does not give off an illumination even while in the presence of light. You can see this because it has a lux of 0, while if I turn it towards the light, the lux will automatically go up. Now that I have 4 milliliters of fluorescein diluted in 150 milliliters of water, I will use my luminescence meter to record the luminescence of fluorescein corresponding to the colored LED light. I will first start off with red, then go orange, yellow, green, light blue, blue, and purple. For our first reading, it has a lux of 5.9 for red. For orange, our lux is 12.4. For yellow, our lux is 31.7. For green, our lux is 47.8. For light blue, our lux is 134.9. For blue, our lux is 76.9. And lastly, for purple, our lux is 89.8. As you can see, through rapid colors change, the luminosity of the solution also changes. So what are the uses of fluorescein? Fluorescence is used in day-to-day life whether you realize it or not. One example is banknotes. Banknotes incorporate security fluorescent fibers of various colors distributed randomly within the paper substrate, and they are detectable solely under blacklight. This helps for a stoppage of counterfeiting currency. Another very important thing is when fluorescence is also used in a variety of food dyes. This is due to the effect that fluorescence increases the viscosity of the surrounding fluid. Lastly, fluorescence is used in a wide variety of medicine. Fluorescence imaging can provide critical information of drug-targeting proteins and also provide valuable information of how the drug is distributed. For example, in the picture of the top right, a wide-field fluorescence imaging of a mouse inoculated in a 4T1 tumor through collection of fluorescence. Thank you for my presentation.